Principles of Inheritance and Variation, Gregor John Mendel, 7 years, that is 1856 to 1863, 7 characters of garden pea. For his hybridization experiment, reason for selecting garden pea or Pisum sativum. Small plant can easily grow in small area like garden, short life cycle 3 to 4 months, clear variation character, easily self-pollinated as well as cross-pollinated. Example, bud pollination. Easily obtained, pure lines, homozygous. A single mating or hybridization can produce large number of offsprings, which minimizes errors. All the traits are characters. Character, any feature versus trait. Trait is a type of character or called as trait. Plant height, tall plant, dwarf plant. Let's understand characters and traits. There are 7 characters and traits. Seed color, yellow and green. Shape, round and wrinkled. Pod color, green and yellow. Shape, full and constricted. Flower color, violet or purple and white. Position, axial and terminal. Plant height, dwarf. Gene versus allele. Gene means segment of DNA which synthesize RNA. Protein, character. Allele मतलब अलग अलग form of same gene. Variant of gene. Allele means allelomorph gene, allotropes, allele. Example, carbon allotropes are diamond and graphite. Gene alleles are capital T, small t, capital R, small r, capital A, small a. Let's understand the characters traits. Characters are controlled by gene factors. Traits are controlled by allele. Let's take a male and female. A male and female donate sperm and ovum, which has 23 N chromosomes. 23 N chromosomes, which both share gene for eye color, for example. So they are homozygous. Paternal one and maternal one has same location of genes. That's why they are called as homologous chromosome. Since they have same location, they are called as homologous chromosomes. Paternal chromosome 21 is homozygous to maternal chromosome 21. Chromosome number 1 contain 2968 locus of various genes. Locus means homozygous and heterozygous, both. Let's understand homologous. This paternal and maternal chromosomes are homologous to each other, but their locus are different. If locus has, but their alleles are different. If locus has same alleles, homozygous, but if locus has different alleles, capital R, small r, it is heterozygous. For example, here we can see capital T, small t, it is hetero. Capital R, homo. Capital small y, homo. Capital A, homo. Capital B, small b, hetero. But these chromosomes, but the chromosomes which are here, they are both homologous, paternal and maternal chromosomes. One homologous pair contain many homozygous and heterozygous pairs. One locus contains maximum 2 alleles. Let's understand differences between homozygous and heterozygous. Homo means same, zygous means gamete, hetero means different, zygous means gametes. Capital T, these are homozygous, capital T, small t, or heterozygous alleles. Okay, alleles, capital T, small t, form one type of gametes, capital T, form more than one type of gametes, capital T, small t. Now let us understand dominant versus recessive. Pure tall and pure dwarf, when crossbreed, removal of anther before maturation takes place. In mature, debagging will take place. After debagging, cross-pollination takes place, and after cross-pollination, rebagging will take place. Dominant versus recessive. Dominant means expressing characters in F1 is called dominant, represented by capital letter, tall character, capital T. Recessive means non-expression character F1 is called recessive, represented by small letter, dwarf character. Example, small t. Let's understand genotype versus phenotype. Genotype means genetic makeup or nucleotide sequence. Phenotype means physical expression of effect of genotype. Capital T tall, small t dwarf, capital T small t tall. Same phenotype tall can have different genotypes. Example, tall, capital T small t. Same genotype can have different phenotype. Let's understand seven characters. Seed color yellow seed crossbreed with green seed. Yellow seed is a dominant one. Yellow seed is represented by capital Y. Green seed is represented by small y. Round seed shape. Round seed crossbreed with winkle seed. Round seed is the dominant one. Round is represented by capital R. Winkle is represented by small r. Pod color green pod, yellow pod. Green pod is dominant. Green pod is represented by capital G. Small pod is represented by small g. Uh, Pod shape, a full pod, constricted pod. Full pod is dominant. Full pod is represented by capital P. Constricted is represented by small p. Flower color violet or purple flower crossbreed with white flower. Violet is dominant or purple flower is dominant. Violet is represented by capital V. Small is white is represented by small v. And here we have last second flower position. Axial position crossbreed with terminal position. The dominant way is axial position. Axial is represented by capital A. Terminal is represented by small a. Tall is represented by capital T. Dwarf is represented by small t. Capital T when crossed, capital T is formed. Small t homozygous gives small t. Capital T small t is tall, which is heterozygous. बर्ड पॉलिनेशन हेट्रोजाइगस स्टॉल को सेल्फ पॉलिनेशन कराने पे कैपिटल टी स्मॉल टी इंटू कैपिटल टी स्मॉल टी आएगा. कैपिटल टी स्मॉल टी कैपिटल टी स्मॉल टी टी कैपिटल टी होमोजाइगस स्टॉल कैपिटल टी स्मॉल टी हेट्रोजाइगस स्टॉल. Let's take a question assertion. Same genotype can have different phenotype. Reason effect of environmental factor. It is the correct assertion and reason. Assertion and reason are true, and reason is the correct explanation of A. Same genotype can have different genotype. Same genotype can have different phenotype because of effect of environmental factor. Let's understand the differences between monohybrid cross versus dihybrid cross. Monohybrid cross, one character for hybridization. Dihybrid cross, two character for hybridization. Monohybrid cross, example seed shape, round seed, wrinkle seed. Dihybrid cross, example seed shape, seed color, both round seed, yellow seed. Round seed and yellow seed. Wrinkle seed, green seed. These are both dominant and these are both recessive characters. Let's understand a pure plant or true breeding plant or homozygous. Tall plant when self-pollinated, capital T into capital T. Homozygous tall into homozygous tall. Capital T is formed. Capital T when self-pollinated. Again capital T are formed. Tall plant when self-pollinated. Heterozygous tall plant when self-pollinated. Capital T small t is formed. Okay, dwarf plant when self-pollinated. Homozygous dwarf plant when self-pollinated. Small t is formed. Small t when self-pollinated. Again, dwarf plant is formed. Small t into small t gives small t which is homozygous dwarf plant. Punnett square is given by British scientist Reginald C. Punnett. Punnett square tells about all possible genetic combination genotypes. True breeding plant is always pure for its characters. On selfing, true breeding plant always produces one phenotype. Recessive characters always pure or homozygous. Dominant characters of phenotype can be pure or impure. Mendel selected 14 true breeding plants. Mendel 7 years, 7 characters, 1856 to 1863 characters versus trait, gene versus allele, homozygous versus heterozygous, dominant versus recessive, genotype versus phenotype, monohybrid versus dihybrid, true breeding plant, Punnett square, and then a first law of dominance or law of paired factors, capital tall into dwarf. Homozygous tall and homozygous dwarf is crossbreed, which gives heterozygous tall. Characters are controlled by factors. Factors occur in a pair. In a dissimilar pair of factors, the expressing character is dominant, represented by capital letter. Non-expressive character recessive, represented by small letter. Capital T into small t. Homozygous tall into homozygous dwarf gives heterozygous tall. Heterozygous tall when selfed self-pollinated, it gives homozygous tall and three and two heterozygous tall and one dwarf character in F2 generation. Tall is to dwarf ratio 3 is to 1 phenotypic ratio, and genotypic ratio is capital 1 is to 2 is to 1, where homozygous tall is 1, homozygous dwarf is 1, and heterozygous tall is 2. 1 is to 2 is to 1. Phenotypic ratio in F2 is 3 is to 1 due to first law of dominance. Mendel obtained genotypic ratio by the help of mathematics. This is an example of the Mendel obtained by the help of mathematics. Homozygous tall and homozygous dwarf, when bred, they give half t, half small t, half small t. When half t plus half t is done, half small t plus half t is done. 1 by 4 heterozygous tall, 1 by 4 heterozygous tall, 1 by 4 heterozygous tall, 1 by 4 heterozygous tall is formed when heterozygous tall is self-pollinated. Heterozygous tall into heterozygous tall, half t, half capital T, half small t, half into t plus half into small t, half capital T into capital T plus half small t. 1 by 4 homozygous tall, 1 by 4 heterozygous tall, 1 by 4 heterozygous tall plus 1 by 4 homozygous dwarf. 1 by 4 heterozygous tall plus 1 by 2 heterozygous tall plus 1 by 4 homozygous dwarf into 4. Capital T plus 2 capital T small t plus small t small t. Genotypic ratio is 1 is to 2 is to 1, as we have got P. Heterozygous round and homozygous wrinkled when crossed Punnett in Punnett square. Let's see. Heterozygous round will give capital R, and homozygous wrinkled will give small r. When in Punnett square, capital R into small r, capital R, small r into small r, small r, small r. So round is phenotypic ratio is 1 is to 1 and genotypic ratio is 1 is to 1. Here we can see some more examples of the mathematical form. When a heterozygous round and homozygous wrinkle is crossed in Mendel's method, capital R will give half r and small r will give half small r. Small r will give half small r, which is equals to r. Half plus half equals to r. Half capital R plus half small r will give, here we have taken this one down here, into r. Half capital R into small r plus half homozygous wrinkle r, half heterozygous tall plus half homozygous wrinkled, half round plus half wrinkle into 2. We get 1 is to 1 ratio. Hence wrinkle and round. Again we take another one, heterozygous wrinkled, heterozygous round and round, pure round. When we get the factors half r, half capital R, half small r, half capital R, half capital R, half r plus half capital R plus half small r into capital R, we get half rr plus half capital R small r, half rr plus half capital R small r. We get homozygous round plus heterozygous round. Genotypic ratio is 1 is to ratio is 1 is to 1. When we take both heterozygous axial positions, capital A, half small a, half capital A, half small a, half a plus half a, half a plus half a, half capital A plus half capital A small a plus half 1 by 4 capital A plus 1 by 4 capital A small a plus 1 by 4 capital A small a plus 1 by 4 small a. When tall and dwarf are crossed, we get tall. When tall is self-crossed with tall, we get 3 tall and 1 dwarf. Here, reappearance of dwarf character. Factors do not show blending. Reappearance of dwarf only possible when factors separate heterozygous tall into heterozygous tall. We get homozygous tall. This is because they are heterozygous. Second law, segregation. Law of segregation. Based on the fact that factors do not show blending, factors segregate at the time of gamete formation. Homozygous will form one type of gametes. Heterozygous will form different type of gametes. Homozygous axial type of gametes is 1 2 power 0 1. Homozygous terminal homozygous type of gametes 2 power 0 equals to 1. Heterozygous axial, hetero means 2, capital A small a 2, 2 power 1 equals to 2. Heterozygous round and LO, heterozygous round and LO, capital R small r capital Y, RY, small r y small r y. These are the two types of gametes formed. The type of gametes equals to 2, 2 power 1 equals to 2. Here we got capital R 1, capital Y small y 2. We will ignore this and we will take this. We have got here two types of gametes, so we will get 2 power 1 equals to 2. Capital R, small y. Here we get capital R, here we get a small y. Since the both of type of gametes are single, we will get type of gametes 1. 2 power 0 equals to 1. Capital R, capital Y. Here we got both the same types of gametes. So we obtain same type of gametes, capital R, capital Y. Type of gametes is 1, which is equals to 2 power 0. Smaller, small y. Here we got two same types of gametes, capital R, small y. So we get smaller, small y. Type of gametes is 1, 2 power 0 is equals to 1. Capital R, capital Y, small y. Here we get four types of gametes, so 2 power 2 square equals to 4. 2 square, 1, 2. Two types of gametes. Like 1, 2, 3. We got here three types of gametes, so 2 cube equals to 8. Here we got three types of same gametes, so here only three types is possible. Only one type is possible. We are not counting number of, but we are counting types. So it is here. It is possible 1, 1, 1. Here we got one type. Here we got same. So 2 power 1 equals to 2 types. Okay. Here we have capital A, same gamete, capital B, same gamete, small b, same gamete, capital D, small same gamete. So one type of gamete will form 2 power 0 equals to 1. Here we got heterozygous, here we got heterozygous. So there are two types of gametes, 2 square equals to 4, right? Yep. Here we have capital A, capital B, here we have heterozygous. So here 2 power 1 equals to 2 types, right? Type of gametes equals to 2 power n equals to number of heterozygous. So here we take two types of gametes. Here 2 square equals to 4, right? When we cross a tall dwarf, we form a tall, which is pure or impure. We don't know, right? So for knowing which is pure and impure, we will do self-pollination. So we'll be forming two tall, three tall variants, and one dwarf. 100% pure conformation. Backcross is outcross. Backcross includes outcross and test cross, I guess. Yep. In outcross, unknown tall, and pure dominant is crossed. So we will get pure tall. When heterozygous tall and pure dominant is crossed in outcross, we get heterozygous. We get heterozygous tall, which is not pure. In test cross, when we cross unknown tall, which with pure recessive dwarf, we get heterozygous tall. In unknown tall, when we cross heterozygous tall with recessive dwarf, we get 50% tall, 50% dwarf. Test cross is done to identify unknown genotypes. Mendel crossed the plant with recessive parents. If unknown genotype is forming one type of gametes, one type of phenotype will be formed. If unknown genotype is forming two types of gametes, two types of genotypes will be obtained. Type of gametes equals to number, or type of phenotype equals to 2 power n. This is one type, so 2 power 1 equals to 2, and this is homozygous, so t, this will be not counted. For test cross, the phenotypic ratio equals to genotypic ratio, capital T into small t, capital T, small t, small t into small t, small t, small t. Or phenotypic ratio is tall is to dwarf equals to 1 is to 1. Genotypic ratio heterozygous tall into homozygous tall, homozygous dwarf equals to 1 is to 1. Heterozygous tall and homozygous dwarf when crossbred monohybrid cross phenotypic ratio equals to ratio equals to 1 is to 1. This is two characters, this is two characters, this is one character. Dihybrid test cross phenotypic ratio equals to genotypic ratio equals to 1 is to 1 whole square, 1 is to 1 or 1 is to 1. Heterozygous, homozygous. Try hybrid test cross 1 is to 1 is to 1 is 1 is to 1 1 is to 1 is to 1 1 is to 1 1 is to 1 1 is to 1. Phenotypic ratio equals to genotypic ratio equals to 1 is to 1 whole cube 1 is to 1 1 is to 1 1 is to 1. Red white red F1 resembling one of the parent complete dominance. Red white pink red white gives pink in F1 does not resemble any type of parent incomplete dominance. Red white red plus white F1 resemble both the parent co-dominants. Incomplete dominance if offspring does not resemble any of the parents. Example Anthurium species or Anthurium majors or snapdragon or dogflower. Second is Mirabilis jalpa or guleban or 4 o'clock plant which flowers at 4 o'clock in the evening. When red and white flower is crossbred, we obtain pink. When pink is self-pollinated, we obtain a red, pure red, and heterozygous pink, and pure white. The phenotypic ratio is 1 is to 2 is to 1, and genotypic ratio is 1 is to 2 is to 1. Phenotypic ratio is equals to the genotypic ratio. Reappearance of red and white flower plant in F2 generation in Mirabilis jalpa or Anthurium species suggest factors do not show blending. When homozygous round and homozygous wrinkled are crossbred, we get heterozygous round. When self-pollinated, we obtain both heterozygous pairs. In the same way, we have here flowers. Color flower when crossed with white color flower we get pink color flower. When pink color flower is self pollinated we get different types of ratios. Red is here pink, as we can see red is 1 and white is 1, but red is 1 and white is 1 and we got 2 pink. Example number 3 incomplete dominance starch grain size in garden P. When large size into small size is done, we get intermediate size. When round and wrinkle is crossed, large, small, round, large, wrinkle, small, we get large and round, and then intermediate and round, and then intermediate and round, small and wrinkled. Capital B and small b control two different characters. Starch grain size or seed size, incomplete dominance. Capital B last side, capital B small b intermediate seed, small b small seed. Seed shape complete dominance. Capital B round seed, capital B small b round seed, small b wrinkle seed.